Hello and welcome to part 3, the last part of that big thick packet that you guys have been working on for, I guess, at least a week now, since last week. So we are moving on. We're on slide 59, okay? And we're going to be talking about, we left off talking about proteins and peptide bonds and two amino acids coming together to make a dipeptide, and then multiple dipeptides coming together to make a polypeptide. And this is how it's done. It's done by the process of dehydration, synthesis of that molecule, in this case a dipeptide, okay? It seems like great big words, so scary. It's not really, right? You know, dehydrated means taking the water out. Piece of cake. The water comes out, okay? Synthesis means making. So we're making a new molecule by taking water out. Dehydration, simply, dehydration is when you take these two amino acids, okay? You got two separate amino acids that you start with the R groups that we talked about last time, that make the beads on the necklace unique from each other. So you can imagine the different colored beads or different shaped beads, okay? When you put them together in different combinations, they have different uses. So again, the R group makes the amino acid special, unique. Put them together chemically, you have a synthesis, it's a chemical bond that they're making called a peptide bond. And in the process, you're removing H2O. You're removing the one water out of this one. And that's the dehydration part. So dehydration is pulling the water out. Synthesis is making that peptide bond. And you've got a dipeptide. And these are the way, this is how you are going to label them, okay? So label both of these on your paper, amino acid. Label that a dipeptide. And then obviously H2O is water. So okay. All right, so I went only one, right? Yeah, okay. So hydrolysis is the opposite. So if dehydration is removing water, hydrolysis is adding that water back in. So it's going to be the opposite reaction. Now we're going to be digesting or breaking down. So when we eat proteins, okay, from plants and animals, we need the amino acids to be used as those building blocks to build the proteins we need for our body. And this is what happens down here. So you have your dipeptide, so it's backwards. Your dipeptide and water, so that's why when you eat protein, you have to drink water, because your body is using water to break those proteins down to make the basic amino acids. If you eat a meal, or if you eat throughout a day and you don't drink any water in some form, juice, soda, whatever, even soda is bad for you, but which is mostly water, then you can get dehydrated yourself, okay? Because your body's using water to break this peptide bond of the polypeptide to make the amino acids. Pretty cool when you think about it. Like lots of things come together in your understanding of what you've learned in the past. So how do proteins differ? Well, proteins differ in the number and kind of amino acids that they contain. That's the kind, in the sequence. So proteins, some of them could be 150, some of them could be 280 amino acids. It's all different numbers and all different arrangements. So many that you could get trillions of different combinations. So here's one example down here. So a polypeptide is only 4 amino acids, right? Of only 4 amino acids, they could be anything from 2 on, right? Has 160,000 possible combinations. So that's pretty amazing. Because if it's 100 amino acids long and there's only 4 amino acids total, they can be mixed up. So you can have 1, 2, 3, 4, 4, 3, 2, 1, 1, 3, 4, 2, and continuing on with the sequences, or you can mix up those 1, 2, 3, 4 sequences. It's 20 times 20 times 20 times 20, okay? 20 to the 4th power. That's pretty amazing. So the vast complexity of life is based upon this ability to make these different combinations. And what's amazing is the DNA, which we'll talk about in the next section right now, is responsible for setting these up in the order that they're in so that they can do the job in your body that they need to do. It's quite a neat system. So how does a protein shape determine its job? So the shape of a molecule, it's kind of like a lock and a key, really, determines how it interacts with other cells. Some proteins will act with your bone cells. Some proteins will act with your skin cells. Some others will act with your intestinal cells and things like that, or your brain cells. And the three-dimensional shape of a protein is determined by that same sequence of amino acids. The different kinds of amino acids lined up in that chain will determine the function of that protein. So remember, each amino acid has a different group, R group, and that makes them unique, gives them a different chemical properties. And in a protein chain, that R group sticks out. That's the part that sticks out of the side, and it interacts with other molecules, okay? Causing in the protein, and it causes it to bend, twist, curl on itself into a very unique shape. Proteins are not just straight long chains. They actually end up being these globular nest of twisted chains together. Okay? So if you look right here, it can meet up with target cells and not disturb or affect non-target cells. So proteins therefore act, because of their shape and because of those R groups that are sticking out, they act on specific cells of your body, which is really cool, that we are programmed to function that way. Our bodies are very specific in their needs, and the proteins help to carry out those specific needs. It's really kind of cool. So we're on slide 63, okay? What are the levels of protein structure? So this is the primary structure, which is just a chain, which is what we've been learning about, okay? It's just the linear sequence of amino acids. So you've got your amino acid, you've got your dipeptide, then you've got your polypeptide structure, okay? Your amino acid sequence. And there's no folding right here. It's just a chain, like a beaded necklace like we've been talking about. And it's made by the genes, excuse me, in your DNA, in your nucleus. And there's a whole process that involves that, which we'll get into that in the future. But your DNA tells your cell what proteins to make, okay? It tells your cell what amino acids to put in order to do the job of the protein. Then, the secondary structure, it starts to coil and make connections, bonds across the coils. So imagine taking the beaded necklace and folding it in half, and then connecting some of those R groups together. You could see, or some of those just H's and O's making what I believe to be hydrogen bonds, okay? Just like in water, when water's negative parts attract to the positive parts, and that's what makes water sticky. Well, the same effects occur here. So you have, yeah, it is hydrogen bonds. I was right. That's supposed to say nearby, not nearb. And so you get these angled structures, okay, called the secondary structure. So some amino acids will hydrogen bond to other amino acids. Then you have the tertiary structure, which now is your true three-dimensional protein. And there's technology today where biochemists can put on virtual reality equipment, glasses and gloves, and they can manipulate and turn these proteins and pull them apart and see exactly what they do. Because eventually, and they're starting to do this now, they can use the knowledge of how these proteins fold and using hydrogen bonds and ionic bonds and other kinds of bonds that are arranged in their hydrophobic and hydrophilic areas accordingly, okay? They can change them and use them to heal sick people. It's really quite amazing. And then finally is the quaternary structure, quaternary meaning 4. And so this is all the different polypeptides chains interacting within the protein itself. So, and this is where that back here, I'll come back to the slide in a second. Right here, this is what's going on here. So certain parts stick out of the protein and can only match up with other cell membranes, particular cell membranes. So that's why they fold like this. And so you've got these different chains with different amino acid R groups sticking out that can react in different ways. So this is an example of hemoglobin that carries oxygen in our blood, and that's a protein. And you see that there are binding sites for oxygen. This is iron. So there's an iron molecule in the protein that binds to oxygen, which carries it around the blood from your lungs to the rest of your body. So that's one particular example of that. So it is time to test yourself. So you are going to do this right after these notes. And we've done this lots of times before. And there's a second page where you have to identify the structures, which we just talked about, using that vocabulary. Now, nucleic acids are the last part, the last macromolecule. So, these are organic, of course, they're all organic. And they contain, therefore, all of the elements that we said, the CHON, the HONC, and phosphorus. One more. There is a phosphorus group that is a very important part of this organic molecule, okay? And it's a chain, DNA. This is DNA. It's made up of a chain of things called nucleotides. This is a nucleotide, okay? It's got 3 parts. Here's the phosphate part. And then those monomers make the polymer called DNA, okay? You get nucleotide down, right? All right. So, and we don't go too deep into this right now because we talk more about DNA in the cell reproduction, which is coming up next, I think, in our units. Okay. So, nucleic acids are important because they contain all of your, like I said, hereditary information, your DNA, and DNA and RNA. So they control the cell, they tell the cell what to do, and they control the synthesis of proteins, which is what I said on the last part. They're the ones that are making those chains of amino acids. So this is your blueprint. This is the DNA molecule, these nucleic acids, stacked into a chain. They control what you look like and how many fingers you have and what color your skin is and whether your hair is straight or curly, and all that stuff, and whether you're a male or a female, and whether you're a human or a cat, okay, or an oak tree. So DNA controls all of that, and and how the body works. Not just what the body is, but how it works. That's the synthesis of proteins. That's what makes the body work. So we're going to review, okay? Ask you some questions. So is this a carbohydrate, a lipid, or a protein? Think about it for a moment. Well, it's got a peptide bond and it's got R groups. So this is going to be a protein or a dipeptide, right? And those two are amino acids with the different R groups. And this is, of course, going to be a certain kind of chemical reaction called, am I synthesizing? Am I making something through dehydration synthesis? Or am I using water in the process called hydrolysis? Right, I'm losing water, not using water. So I am dehydration synthesis. That's what this is. If water comes off, it's dehydration synthesis. Okay, is this a carbohydrate, a lipid, or a protein? Instantly, you should see that hexagonal shape. Looks kind of like a stop sign, but not quite, because stop sign's octagonal. That's 8 sides. This is 6 sides. You know that this is some sort of sugar, so it is a carbohydrate. And you've got 2 of them here that are needing water to break apart. So the water is being added back into them, and then now you've got two single units, okay, which are monomers. So is this synthesis or hydrolysis? This is not synthesis. It's breaking apart, right? This is hydrolysis. This is adding water. It's not dehydration. You're adding water. Remember that. And so this is digestion, breaking down foods to the basic parts. And naming the molecules. This is your disaccharide, 2 sugars, right? And then these are your monosaccharides. Is this a carbohydrate, a lipid, or a protein? Do you see any R groups? No. Is it hexagonal, like glucose? No, so it must be a lipid, because these are your fatty acids and your glycerol, and this is called a triglyceride, okay? So you got your fatty acids, 3 fatty acids of glycerol, and your triglyceride, 1, 2, 3, and your glycerol, triglyceride. And then, of course, 3 water molecules. So is this synthesis or hydrolysis? This is synthesis. I'm putting together smaller units to make a bigger unit. Synthesis. Dehydration, synthesis. The water leaves. Dehydration. Obviously, this is going to be on your test. All of these things. So you got to know the differences between these macromolecules, okay? And I'm going to give you some more practice in the future. And then you have final 4 questions. These are in your notes. What are the 4 classes of the organic molecules we learned about? why are they all considered organic? And then this one, we didn't really talk about it, so you have to infer based upon your knowledge why it would be bad to eliminate any one of these from your diet, okay? If you just ate carbs, or you just ate protein, or you just ate fat, okay? Well, what would happen to you? So look it up if you need to, okay? But I am going to be checking your notes, and I'm going to make sure that you have those done. So you can use some class time now, but we have another assignment that we're also doing on the board and Google Classroom, so check that out.